This might be bad though, Turgus. This is not Gucci Gucci Goo. This is the science news story number 3 of the night, Turgus. And this is something that we did not think would ever be an issue because of how CRISPR genetic engineering works, but it turns out that it is, and this has relatively high consequences, possibly, potentially, maybe, on using CRISPR genetic engineering as a therapeutic. So, why does this work the way, like, what is it meant to be like? How does it end up working? What is the point of it? Citing your genome. You can cut out that mutation and fix it. And the idea is that it's very, very specific and it shouldn't have off target effects, meaning it should only be cutting at the site that you tell it to cut. It's been used in a ton of different organisms. It's been used in a bunch of different disease states. In theory, it should be able to be hit any single disease state and fix it. The way that it repairs, it'll show you in this video, is called a homology-directed repair. So, Didi, what that means is you take your double strand of DNA and you break it. When you break it, you provide that breakpoint. Right at the break site, you give it something to integrate into that breakpoint. So normally, if you break a piece of DNA, your cells will try to fix it. There is machinery in your cells to fix broken DNA. Awesome. But if you give it something, it might even insert an element that it's trying to repair. So if that extra element floats in at around the break site, it might get integrated into the existing piece of DNA. And that's one way DD builds. Let's say if you have a mutant copy of a key gene, you break that mutant copy, put in the correct version of it, and now you got a functional gene. Easy squeezy, lemon peasy, right? Well, let's see how the system works, then I'll tell y'all why it's maybe not easy squeezy, lemon peasy. So let's get to this new paper again published in the journal Nature Biotech. Nature is one of the top journals. Nature Biotech is a subsidiary of Nature. So it's not as big, but it's still a pretty good big deal to get in here. And so the goal of this is actually like the researchers actually we're trying to make a therapeutic. And trying to fix certain disease states. And so the molecule that they're using is called AZD7648. And what this molecule does, it blocks the ability of the DNA in the cells to quickly fuse back together. So remember how we talked about that there's a couple of different options. One is if you break the DNA, you want to be able to insert something into the DNA itself, like your fixed new gene. That sometimes happens, right? But other times, the DNA just fuses back together. And remember, Sam pointed out, if the DNA fuses back together and you make a mistake, that could still be very bad. And we don't want that to happen. And so they made this molecule, this AZD molecule, that slows down the process of the two broken strands reconnecting. So it increases, hopefully, the likelihood that whatever sequence you want to put in is the sequence that's actually put in. Does that make sense, y'all, of what the point of the molecule actually is? Because it's a really, really important molecule, and it could be really, really helpful then for treating these disease states. Use it in humans should be initially with a no safer option. So this is a risk, Sam. The paper is trying to identify that. I understand where you're coming from. Totally. Yeah. So it turns out though that there is side effects to this. So again, the goal of this molecule is to have there to be that breakpoint in the DNA from Cas9, and then you're inserting the genetic element that is correct. However, it turns out that by using this molecule, a lot of the cells that were in the mix led to big genetic changes in parts of the genome that were not expected to be edited. And what was the edit, Sam? Here, this edit is probably worse because it led to deletions. Even whole chromosome arms were lost. So when they looked at and here was the catch, right? If they looked at the gene that they were trying to edit, that was edited beautifully. 100% of the time, Sam. You're cutting here. Every time it inserted the genetic element they wanted, it didn't bind back together. That means it's doing a great job, right? Yeah. I know, Shadier Tree. Hi, welcome in. But then the catch became they were not, it was, if you look elsewhere. And here's the thing. Oh, yeah. Well, but Sharky, here's the financial issue, right? If I were able to fix a gene in your cells, it is, from a financial perspective, much easier just to check and make sure that the site that we want to fix is fixed rather than checking your entire genome. And so here, they actually go and check the entire genome, which for that, just from a study perspective, you need a lot of money because you need a sequencer. You need a lot of replicates, so that's a lot of different sequencing experiments to do. And then you need someone to be analyzing the data. And so, again, what they find is that there's these global mass, like genetic changes, and you don't see those if you just look at the gene that you're trying to edit and the regions right around the gene that you're trying to edit. So it's like, if you're only looking for what you're studying, that's all you see. What it looks like physically. So Casey, good thing they did not put this in a person. I will show you all the figures. We will go into the figures in just a second. So in terms of physically, they did have some like DNA images that we were looking at, so you can see what it looks like in terms of how the cell is coming apart. There was no animal that it was put in, so like no animal or anything underwent this kind of breaking point, but it's surprising. And actually so Shark Geek, it's so narrow that they haven't yet looked at all of the genome. They just looked at like the really big parts where, like, oh god, something is visibly wrong here. Where they're trying to find a region of the genome and they're like, wait a minute, this region is missing. And they're like, well, let's what is going on here? They even said in the study there's probably other regions of damage that they have not yet found. And so that's like a next thing to look at, right? That they are pointing out is like, okay, even in this system, we need to do an even deeper look before this molecule is being utilized. One question which I thought was really interesting that they framed in the study is why did they even look for this? Right? Like, why it works so well. Why would you ever do this? Right? Like, you looked at the region of interest. It works great. Why would you look at something different? The PI, the principal investigator, was like, actually, we thought it worked too well. Yumi, thank you for that follow. Welcome in. They were like, it worked too well. It works too well here. Let's check this out. And they actually did mention, even in the paper, they said, quote, this is a major setback. And they even used the phrase catastrophic side effects of using this small molecule. So it's not using CRISPR, it's using that small molecule, this AZD, that's meant to promote the two strands of DNA not binding back together right away, but letting you insert something, right? So the idea is, if you have a patient with a disease state and you want to fix it, you'll break the DNA, you'll insert the correct version of the gene, and then the patient can go on and be healthy, right? You don't want the DNA that you're breaking just to reconnect, because you want to maximize the likelihood that you're going to help the patient. And it seems like this small molecule goes loosey-goosey and causes a bunch of damage elsewhere in the genome. Digital coding were one thing. So yeah, it's just surprising, Didi, like when you do CRISPR itself without this molecule, right? It goes to its target site really, really well. That 25 base pairs that you give it for the guide gives it that specificity to go to the correct site. It seems like just these other small molecules that are being added in to kind of make the process better or more efficient, those small molecules aren't specific enough, right? If this molecule is then decided to free flow, go elsewhere in the genome and cause there to be these breaks, that's really less not good. It's far from ideal. And there's just no way of, we think, fixing that, at least not yet. So what are the other takeaways before we go just into the meat and potatoes of the paper? Um, it's not the end of anyone trying to use this, right? Of the CRISPR technique to fix this disease state. It's that they just need a different molecule to promote the end joining, where you allow an insertion of a genetic element. Um, if there are other candidates of small molecules, they need to look for this as a side effect, right? Because they didn't know that this was a side effect. Also, Sam might be like, hey, didn't we talk about a treatment for sickle cell anemia that used CRISPR? What happened to those patients? Is that patient where we use CRISPR in sickle cell, are they just nerfed now? Did we just ruin their genome? Don't worry, Sam. We did not. Because the experiments and treatments that have been used for patients with sickle cell, the small molecule AZD was not used, which is great. This was meant to make things easier, but then it was identified, no, it actually makes things worse with the additional breakpoints. Yeah. They were, the feeling I think they had was they were so hyper focused, right? Because the only goal has always been just to fix this region of the cell and this gene in the cell. What ends up happening if you don't just fix that? You don't even think about what the rest of the region is, because the CRISPR works great. We've already tested the whole genome when you do CRISPR, and the genome is great. Everything's fine. Why would you? You're just making a process better. GG, Scully. And it turns out you don't. And so this is kind of the reasons now to look for it. So these are the guide RNAs, the reading sequences, and we're just getting a bunch of measurements, and here is kind of the nice part. So in sequencing experiments, you get what are called reads. Reads are segments of RNA that correspond to the whatever you're amplifying. If you have everything, the whole genome covered, that is your reads that are covering the whole genome. So you've mapped out every single element of the genome, and that's being captured in your data analysis. If you're missing reads, it's either a technical issue or because you have deletions. And here you see with a deletion, you have far less reads that you're getting. And so that was their first show of wait a minute. It's not just that we forgot how to sequence. Casey was wondering about if there was imagery behind it. So Casey, the figures that are meant to illustrate this are here in panel G. And let me just tell you of how they say it. So this is called a Circos plot of the sequencing. And so I would have liked to have seen a karyotype. A karyotype is what you see when, let's say, a patient has trisomy 21. That's like an image of the chromosomes in your cell, and then you have a third chromosome 21, and you can very easily see it, and it's beautiful. That's not the kind of image that is here. Instead, this is a visual plot meant to illustrate um, what is happening within the cell. And the way that they illustrate it is there's 3D interactions between different regions of the chromosome, or different chromosomes throughout the nucleus of the cell. Um, as in this 3D space, the DNA interacts in different ways. If there are regions missing, the DNA cannot interact. So normally, image here, this is your control image. This is when that you've inserted a breakpoint at a direct point in the genome that you know is broken, that HBB, that's where you've broken it. You've inserted something different, and now normally where there could be an interaction between chromosome 11 and 6, there's not one. That's what you're demonstrating here. The neighboring image, huge. Look at it. So you're having now issues not just on an interconnectivity on 11 and 6, but 11 and 12 that should interact, 11 and 17, and 11 in the X chromosome. So those red dots, this graph doesn't give you the intensity of break. It just tells you where there is now a mistake. And so that could be in the form of missing a massive arm deletion, or it could be a smaller deletion as well, but it doesn't the point is there shouldn't have been a breaking point on any of these other regions. Right? And so that's giving you a direct image of what's going on when you add in this small molecule that you're wanting to change, right? You know that if you change it, it stops the interaction between chromosome 3 and 11 at that certain site. Fine. We know that's what we're doing, where that's what we're putting in intentionally, right? Here, if you then add in another reaction, a different chemical reaction at a different location, you add in that AZD, all of a sudden it's hitting other chromosomes as well. So these two figures, what they're illustrating is that this isn't just an isolated incident. This isn't just a chromosomal change at one site due to one time event. Because, right, Casey, the argument could be is like, well, the whole thing that you just found is just a byproduct of you targeting this one gene. If you targeted a different gene, this wouldn't happen. So this is only a bad thing in this one scenario. But they're showing here that no, that is not the case. It is not just a bad. And here's a third one. So Casey, this one was like the creme de la creme of bad. Here, they're hitting the gene GAPDH. GAPDH is a chromosome binding protein. That's important for chromosome pairing, which is important in cell division, whether it be mitosis or meiosis. So it is a fundamental thing. And actually, there are human diseases where this is mutated. This one didn't have any interaction anywhere else. You're just mutating GAPDH on chromosome 12 without the small molecule. And then if you add in AZD, look at it, almost looks like a volcano just erupted. All these interaction sites now are broken across many different chromosomes. And so, right, Casey, it'll happen every time, but the intensity is different every time, right? So it's not only that it's causing chromosome scarring when you use a small molecule, it's causing chromosome scarring in a not predictable way, right? Maybe it's predictable if you know what gene they're changing, but Casey, there's no way for me to test every single gene, go on in there, and then anticipate it, because then this being an easy, molecule to make things easier, it no longer makes things easier. It's actually a really frustrating molecule now. It's causing there to be a lot more issues than there are solutions. And yeah, they only changed the one gene, right? And they're changing either the addition or subtraction of the one molecule, and there's this much of a difference. Again, I wish there would have been like a karyotype. I know there's no real reason to have it. Um, beyond me thinking it would be cool. I don't think it was in the supplement either. Yeah, they're giving us the sequences. This is great for anyone who obviously wants to replicate the study. They're giving you the sequence of what is the guide RNA that they used, what kind of cell type they put it in, what the length of the guide RNAs were, what the sequences are, what the mutation type ended up being, and then here are all the different mutation types, Casey, that they found that were caused as a function of this small molecule. So you can look at. So again, it's really cool to have been able to demonstrate all of these different avenues. They also showed all the materials that they got, where they got from, with a catalog number. Look at this beautiful table. Sam, if you and I wanted to order the same things and do it in our garage, I mean, Sam, we couldn't because it would be cost prohibitive. But God, by golly gosh, if we had unlimited coin, we could do it. It's again, we can't because we don't have the money for it. But the reason I point this out is because we've had plenty of papers where we haven't actually had all these things listed out nicely.